And we will turn on to Tessa Bertozzi from the UK.
Hello, my name is Tessa Bertozzi, and I'm a PhD student at the University of Cambridge in the UK. So my research is in the field of epigenetics. So what does that mean? If I were to look into the nucleus of one of the cells of this mouse, I wouldn't just see naked, free-flowing double helices. DNA is actually wrapped around proteins called histones. And the DNA strand, as well as these proteins, are decorated with chemical tags. And these chemical tags are able to turn, or at least contribute to turning some genes on and some genes off. So even though every single cell in this mouse has the exact same DNA sequence, each cell type has a different combination of these chemical tags, which help contribute to cellular function. So we call these tags epigenetic marks. Now, the epigenetic mark that I study is called DNA methylation. DNA methylation attaches directly to DNA, specifically to the cytosine base, or C base, also called C base, and it is normally associated with gene silencing. Now, unlike DNA, which is quite stable, DNA methylation has been shown to be sensitive to environmental changes. So now that I've given you a bit of an introduction on epigenetics, I'd like to tell you about quite an important mouse model in the field. So every single one of these mice is genetically identical. But the reason they have such variable coat color is because they're epigenetically different. So they all carry the gene that codes for yellow coat color. And in yellow mice, this gene is constantly turned on, so the mouse looks yellow. Now, when there's methyl groups, so DNA methylation I was talking about earlier, near this gene, the gene is off, and the mouse looks default brown. Now, this is quite cool in and of itself, but one of the reasons it's such a popular model is because a yellow female is more likely to produce yellow offspring than a brown female is. Now, I've told you they're genetically identical. So this means that more than just DNA is being passed on to the next generation. So this is quite a new concept. Up until recently, we've thought that we only really inherit DNA from our parents, but this might not always be the case. And this is known as epigenetic inheritance. So what we were interested in determining in our lab was whether there are other regions in the mouse genome that behave in this way and show variable methylation levels between genetically identical mice. And if they do exist, I wanted to figure out what their inheritance patterns are and whether they're susceptible to environmental changes. So we used genome-wide methylation datasets to try to find regions that behave in this way. And we found over 100 of them. So the graph you're seeing here on the left shows the data collected for one of these regions. So each line represents a single mouse. All of them are genetically identical. And on the x-axis, you have the cytosine base. And on the y-axis, you have their methylation levels. You can see that there's a wide range of levels across these individuals. So to figure out what's happening in the next generation, I selected highly methylated females and lowly methylated females, and I looked at what the methylation level of their offspring was. And as you can see on the right, regardless of whether the female is highly methylated or lowly methylated, she can produce offspring covering the whole range of methylation levels. So this epigenetic variability we're observing gets reconstructed from generation to generation. That said, that doesn't mean that a highly methylated mother isn't more likely to produce highly methylated offspring than a lowly methylated mother. So to test this, I had to look at tons more mice and figured out that for the most part, there is no memory of parental methylation state. So arguing that epigenetic inheritance is actually quite rare. I did find one region, however, that did show an effect. So each dot here represents a single mouse. And as you can see, offspring from highly methylated mothers are on average more likely to be highly methylated than offspring from lowly methylated mothers. So it's rare, but it can happen. And finally, I mentioned that there's this idea that epigenetic marks might be susceptible to environmental changes. So I decided to look at folate deficiency as an environmental exposure. You might have heard of folate, also known as folic acid, because it's a supplement often given to pregnant women to prevent birth defects. So I looked at normal mice and folate deficient mice and assessed what their methylation levels were at these regions. And what you can see here is that there's a drastic decrease in methylation level in the folate deficient mice compared to the normal mice, suggesting that these regions are susceptible to environmental changes. So I'm now looking into other environmental exposures, including diet and toxins, to figure out whether this phenomenon is more widespread. And with that, I will take questions.
Thank you very much. Okay, so where do we have a question? Maybe about the motivation of the research, or... Yes, here.
Thank you for the presentation. Is this research applicable to humans as well, or is it just is it just for mice?
Yeah, that's an excellent question. So yes, the answer is there's lots of research happening in the field of epigenetics in terms of human biology. This research in particular is difficult to do in humans because you need genetically identical individuals, right? And of course, we don't have genetically identical humans that we can look at, nor would this be ethical. So for this kind of study, we really need to use lab mouse strains where we can control genetic differences. But yes, and there are groups out there trying to find regions that behave in this way in humans.
There's one more question here. See that?
Shouldn't twin work like twins, because they should be genetically the same?
Yep, so absolutely. So actually that's the one exception where you can use humans to look at this. And there have been studies, looking at how different the epigenome is of twins. And there have been found to be differences. So as twins grow up, they grow up in increasingly different environments, and so you'd expect there to be some changes. And yes, for these regions, we're looking for ranges, and that's sort of just 2 individuals. But yes, and that's ongoing research being done by other groups.
And how many mice did you use?
Oh, quite a few. In each of these, in the original experiments, it was about 8 to 10 was the number we wanted to use to show that there was true variability between them. And as you can see, for these kind, it's up in the double, triple digits.
Thank you very much.
No problem.
There's one more question over there.
Hi, okay. So what I was wondering was if epigenetic inheritance, if you could apply it to humans, so this is basically very far ahead. If so, if it could be applied to medicine in any kind of way.
Yeah, so actually, epigenetics as a field is already being applied in medicine. So there are already drugs on the market that use epigenetic mechanisms to sort of help control gene expression or alter gene expression. The inheritance part of it, there's a lot of interest in determining whether, what happens in one person's lifetime affects the next generation or even generations after that. But as you said, it really is very early days. It's difficult to even establish if it happens at all in humans. So we're pretty far away from that, but there's lots of interest in the topic.
Thank you.
You're welcome.
There is one more question over there.
At the picture with the mice with different colors of fur, did you yourself or like in lab try to control the methylation of them, or was this just an example of how they can develop differently?
Yeah, great question. We don't work with that mouse strain in our lab, so that was actually discovered quite a few decades ago now. But there have been lots of studies on that mouse model where they have some kind of environmental shift, so increase the specific components in diet or something like this, and that has been shown to shift the coat color distribution. So say in a normal context you have a certain proportion of yellow mice and a certain proportion of mottled ones and a certain proportion of brown ones, and you can actually shift the whole population towards yellow or towards brown, depending on environmental changes. So yeah, it's been done.
Okay.
Okay, any other question? Sorry, down here.
Can you use epigenetics to get glow-in-the-dark mice?
So glow-in-the-dark mice are actually made by changing the DNA itself.
Yeah, okay,
so
it's DNA.
Yeah, so those often you direct and change the DNA. But you know, now there are technologies where we can maybe direct and change the epigenetic marks at certain regions.
Yeah, but then shouldn't you be able to deactivate the expressions in the DNA to get glow-in-the-dark mice to be like white or something?
Well, so now there are already inducible systems that allow you to have the mice that don't express the gene you're talking about, and then you can activate it. So since we already have a means of doing that, there's maybe less drive or interest in using epigenetic mechanisms, but...
Very cool.
Thank you.
Okay, there's just, one question.
You said you could change the color of the fur of the mice depending on environmental changes. How would it look on humans?
Yeah, so since we don't have an exact same situation in humans yet, at least, it's a bit difficult to predict how that would work. It certainly wouldn't be fur, but it might be some other gene. But yeah, who knows? Hopefully there's something similar.
Thank you.
Yeah.
There's one more question up there.
Okay. This is a more general question. What was your main motivation to research your field?
Let's see, there are quite a few. Weirdly enough, in high school, our my biology teacher gave us extra reading one day, and those mice were on the front page of that article. And I thought, you know, what are these yellow mice? And that kind of got me inspired to study biology. And then I kind of forgot about it for a while, went to university and studied molecular biology, found out about epigenetics again in my final courses, and then decided to apply for a PhD within the field, and have now ended up working on something quite relevant. So what you learn in high school matters.
Thank you.
You're welcome.
Great. Okay, is there any other question? Yes, down here. Okay, this is like a very popular topic.
That's great.
Do the experiments cause the mice like physical pain?
That's a very valid question, and it's an important one for all future scientists to think about. So whenever you want to do research that has any kind of human health implication, or even relevant to mammalian biology, you have to use a mammalian system, right? So we use mice, and there's tons of regulation in place to make sure they don't suffer any pain. So they have a great cage with things that can run around in, and water and food, and at the time when, and then they don't, the way that we end their life is not, does not inflict any pain at all.
Okay, great. Thank you so much. Okay, wow. Thank you.